So today we're going to talk about DNA replication, which one of the main things I want you to remember about DNA replication is it's a rare occurrence. Well, when I say rare in the lifetime of a cell. It only happens when mitosis occurs. You only have this happening once in a cell's life cycle. It's not like transcription where you're going to be making it constantly. You're constantly going to be making mRNA to make different proteins. This is a rare occurrence. And it is semi-conservative, meaning that actually by the time that you make a new one, each strand, half is one of the original strand and half is new. We'll talk about that more in a different video. But semi-conservative DNA replication. So the thing that makes this hard is the fact that the DNA polymerase, which is actually this, only functions and only can build DNA strands in the 5' to 3' direction. So, as always, don't try and overlearn it, just understand it. So let's count the carbons. So this is the 5th, so this is our 5' end, and this is our 3' end, up and down. So when I say it only functions in the 5', 3' direction, basically what I'm saying is you can only add another nucleotide and form another phosphodiester bond here. So you can't do it at the top end. So that will then form a phosphodiester bond, actually forms the hydroxyl group, but you can't do it this way. The polymerase doesn't fit on like that, so it has to go 5' to 3'. Same is true of the RNA polymerase, but we'll explain later why it's more important here. So that's what DNA polymerase does. This is what DNA replication is. It's just reproducing another strand. However, it gets more complicated. It gets infinitely more complicated. When you have DNA, you obviously have the 5' and 3' end of the original strand. So the green is the original strand here. When you split it with a helicase, which breaks the hydrogen bonds, so the helicase unzips that original parent strand of DNA, it breaks them apart and it creates this thing called a replication fork, which is this area here, basically where the helicase has got to. And now, if we look at how these things are going to align, I've actually drawn out what these nucleotides will look like. So when you're building the complementary strand to the 3' end, actually the nucleotides are arranged in the correct way to grow towards the replication fork. You've got this OH here, so when you want to go and add another one, your DNA polymerase can do it towards the replication fork. And actually they use triphosphatidic nucleotides, but that's another story. So you then form that, and you are growing your nucleotide chain. And you're replicating your DNA, where this will say this is an A, there'll be a T on the other side, on the green one. And say this is a C, there'll be a G over here. So you're getting your hydrogen bonds forming, perfect, and then this DNA polymerase will come in and form the sugar phosphate backbone. It's more complicated on the other side, because where this is the 5' end, where it would be the 3', where it's the 3', what you'd actually have is something that looks a little bit like this. Dum-de-dum-de-dum. Okay. And say, I don't know, say that's an A on the green, and then over here is a T. You can't add, we've just said, this is the 5' end, you can't grow the chain towards the replication fork. So you end up having to work backwards. So you grow from here, and this way it is actually arranged in the correct manner. So now we've got this. Let's draw the little thing on, the ribose, or deoxyribose, this is DNA, and there's an OH group here. And now we can grow that way, because we can grow in the 5' to the 3' direction. And that's fine, that'll work. And if we just let these things run, if we just let this run, this strand would grow like so, and this strand would grow like so. But this is where it becomes not confusing, but this is how do we get these weird fragments, and what's the leading and the lagging strand? I remember those. What ends up happening here is the DNA pol will chase the helicase on the leading strand. So When this DNA helicase and gyrase working together, gyrase is the one that untangles it, you don't need to know it, but I thought I'd mention it, sometimes it's on mark schemes. DNA pol will basically chase this helicase, and as the helicase unzips it, the polymerase will follow it. On the other strand, it's not so simple, because it's growing in this direction away from the replication fork. So by the time it's done all of this, it's got no choice but to go back to the replication fork and work its way backwards again. So actually on the lagging strand, the reason it's called the lagging strand is because it's literally slower. Think about playing a video game or something, it's slower, so you're lagging behind. It's because it's got to constantly keep doing this and going back to the replication fork and then back to the replication fork. So that is why DNA replication is so confusing, why you have these lagging strands and leading strands. It's all to do with the fact that the DNA polymerase can only grow in one direction, and the DNA helicase is doing its thing up ahead on the chain, and therefore you have one strand that can follow the helicase and one which keeps needing to go back to the start where the replication fork is. So you've got your leading and your lagging strand.